Hi, I'm Rochelle Crosby Watson, and we're back again to talk about excitation-contraction coupling and how this process regulates force production. So, let's start with a case study. A soccer player is allergic to dairy products, and he's getting ready for a big soccer game, and he decides to not eat or drink anything on the day of the game. Doesn't sound like a good idea. Halfway through the game, he falls over in terrible pain, and he also has these horrible skeletal muscle contractures shown here. They look awfully painful. So what's causing this? Well, let's take a closer look. Let's dissect out the process of an individual skeletal muscle twitch. And in an individual skeletal muscle twitch, the skeletal muscle is stimulated to contract. There's a latent period before the skeletal muscle achieves its maximum force contraction, and then there is a relaxation phase. One analogy that I like to use here is your regular gas stovetop. You know that when you're turning on your gas stovetop, you hear a spark. There's a latent period before, boom, the gas catches fire. So that timing is very similar for skeletal muscle contraction. There's a spark or a stimulus, a latent period, contraction, and then relaxation. The process that regulates the skeletal muscle contraction is a process called excitation-contraction coupling. And it's really a very elegant process, and we're going to go over the basic elements here. So as you know, skeletal muscle is innervated, and that site of innervation is called the neuromuscular junction. The sarcolemma membrane at the neuromuscular junction is highly folded, and those folds are enriched with acetylcholine receptors. So upon stimulation, the motor neuron releases acetylcholine. The acetylcholine travels across the cleft and interacts with the acetylcholine receptor. That sets off the electrical stimulation that travels down the sarcolemma, all the way down the T-tubules. Remember that the T-tubules are a structure, a membrane invagination of the sarcolemma that goes deep within the skeletal muscle belly. The T-tubule communicates with the sarcoplasmic reticulum. And in fact, the sarcoplasmic reticulum is a membrane sac within skeletal muscle that stores calcium. And the T-tubule and the sarcoplasmic reticulum communicate with each other by virtue of these two large complexes of proteins. We're going to see it on the next slide. The proteins are called the dihydropyridine receptor, which is a voltage-activated channel, and the ryanodine receptor. The ryanodine receptor sits in the sarcoplasmic reticulum just at the interface with the T-tubule, and the dihydropyridine receptor sits in the T-tubule juxtaposed to the ryanodine receptor. So these protein complexes are found in two different membrane structures, and they're communicating with each other by virtue of protein-protein interactions. So the ryanodine receptor receives a signal from the dihydropyridine receptor. Hey, I'm now voltage activated, and that causes the sarcoplasmic reticulum to release calcium inside the sarcoplasm, and that calcium activates the actomyosin interactions, and it does so by interacting with troponin C. Troponin C binds to the calcium that's released from the sarcoplasmic reticulum, and when calcium binds to troponin, it causes the whole troponin-tropomyosin complex to become very flexible, okay? So the troponin-tropomyosin complex is very flexible. It's rocking and rolling over the surface of the actin filament, okay? Now, what's happening when it does that? What's happening is that it's exposing its myosin-binding sites. So normally, in the absence of calcium, the tropomyosin and the troponin are covering the myosin binding site on actin. but in the presence of calcium that causes troponin, tropomyosin to undergo conformational changes. Those conformational changes cause this rocking and rolling, this flexibility that now allows myosin the opportunity to bind and grab hold to actin in its specific myosin binding site. And of course, we've already seen what happens when actin and myosin are interacting and undergoing the cross-bridge cycle. So now we're looking at the same process, just a different illustration. And we're going to look at the flow of the calcium molecules and the interaction between the dihydropyridine receptor and the ryanodine receptor. So when skeletal muscle is stimulated to contract, that action potential travels down the sarcolemma membrane and down the T-tubules. Those T-tubules reach deep within the skeletal muscle belly, allowing the skeletal muscle to contract all at one time and to allow rapid transit of that signal. So the dihydropyridine receptor is communicating with the ryanodine receptor. Remember, the ryanodine receptor is a complex set of proteins in the sarcoplasmic reticulum. When the dihydropyridine receptor receives the voltage activation that causes the release of calcium from the sarcoplasmic reticulum into the belly of the myofiber, where those calcium molecules interact with the troponin tropomyosin complex, causing this flexibility, allowing now the myosin thick filament, shown at the bottom of the illustration. The myosin thick filament now sees that its binding site on actin is revealed. It reaches up, grabs, and undergoes a power stroke, as long as there is ATP around. So that, in a nutshell, is excitation-contraction coupling. So when you're studying excitation-contraction coupling, it's important to think about and identify the location of the various players in EC coupling and what their function is. So this particular study question just lists out some of the major players, and it would be a good idea for you to think about where they're located and what their specific function is. So, a 1-minute write. List the order of events from the beginning of the motor neuron innervation to skeletal muscle contraction. What are the precise events in excitation-contraction coupling? So, we've written out some of the major events that take place in excitation-contraction coupling, and you can compare this answer with your own. So, let's go back to our case study of the soccer player that collapses halfway through the soccer game. What do you think is the cause of these skeletal muscle contractures that he is experiencing? Well, most likely he is having an absence of calcium. Remember that he's allergic to dairy products, didn't eat anything or drink anything the day of the game. Also, he may not have ingested enough energy in the form of ATP to release those cross bridges so that actin and myosin stay bound to one another in a contracted state. In other words, there was no ATP to allow for release of those myosin heads, which is a common state called rigor mortis. Now, let's go back to our muscle twitch. This is the profile of an individual muscle twitch that happens from a single stimulus. What would happen if instead of giving one spark to ignite the gas, we gave multiple sparks, pa, pa? What about multiple sparks, pa, 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 right on top of each other? What would multiple rapid stimulations do to skeletal muscle twitch production, to force production? What would it do to the intracellular calcium concentrations? Think about it. So here's a 1-minute write. Predict the cytoplasmic calcium levels during a single twitch, summation, and tetanic contraction. Draw the calcium concentration and the force curves on the previous diagram. So the answer is here. Multiple stimulations cause a release of calcium that tends to increase because not all of this calcium is returned back into the sarcoplasmic reticulum at the end of contraction, that leads to a greater and greater buildup of calcium inside the cell. That increase in calcium upon each additional stimulation causes an increase in the overall force production. So this is one way that muscle has to regulate the force production abilities, simply increase the amount and frequency of stimulation. So while we're on the topic of skeletal muscle force production, it's prudent to mention that skeletal muscle can be divided up into two major types. Slow or type 1 fibers that are built for endurance, and fast or type 2 fibers that are built for power and speed. And these skeletal muscle fibers have different properties in their metabolic profile, their resistance to fatigue, their contraction speed that allow them to do different functions. A whole skeletal muscle fiber is not entirely slow or entirely fast. Skeletal muscles are really a mixture of slow and fast, and the relative percentage of the slow and fast can vary depending on the overall function of that skeletal muscle. So take a look at this histological stain. This is a skeletal muscle cryosection, and I hope you can see that the cross-sectional diameter of the fast fibers is much larger than the cross-sectional diameter of the slower fibers. We've learned through studying electron micrographs that a majority of skeletal muscle is made up of those gorgeous myofibrils. And those myofibrils are what lead to force production and actomyosin interaction. So it makes sense that the larger fibers would be the faster fibers and that the smaller fibers will be the slower fibers. In addition to those other characteristics, slow and fast fibers actually express different myosin subtypes. And we refer to those as the slow myosin heavy chain and the fast myosin heavy chain. And those myosins have different properties in the actin cross-bridge cycle. Also, suffice it to say that a motor unit can be either all fast or all slow. So in other words, the skeletal muscle fibers that are connected to each other by virtue of the innervation of the motor neuron, all those fibers are going to be of the same type. So when you're thinking about skeletal muscle fast and slow, that can dictate the regulation of skeletal muscle, and it can dictate the overall function of skeletal muscle. I bring this up now because when we start to get into some of the details of the mechanism of disease, of muscle diseases, the distinction between a slow fiber and a fast fiber will become very important. So, thanks for tuning in, and we'll see you at the next lecture.